Ever stop to think about the hidden world living right on your skin?
It's pretty wild, isn't it? A whole ecosystem.
Totally. It's a bustling microscopic place, and sometimes, certain residents can cause a bit of a kerfuffle, like Cutibacterium acnes, a big name when we talk about acne.
Right. C. acnes often gets blamed.
Okay, let's unpack this.
It's really fascinating that something like acne, which we often see as just, well, superficial, actually involves this incredibly intricate dance between our skin and the microbes living there.
Absolutely. And in this deep dive, we're doing more than just looking at the science bit. We're also diving headfirst into the really creative world of the Cütie Catcherz AR comic series.
Oh, interesting connection.
Yeah, we've got some recent research on Cutibacterium acnes that basically lays down the scientific groundwork for the comic's super imaginative stories.
So real science feeding into fiction.
Exactly. Think about it. Genuine scientific findings sparking these, incredibly inventive, metaphor-rich narratives. So today isn't just about understanding what might be behind a breakout. It's about seeing how the detailed world of microbiology can actually ignite creativity and storytelling.
And when you connect that to the bigger picture, you realize that understanding the basic science can actually make even these fictional stories feel, well, more resonant, more insightful.
And that's really what we're aiming for with you today, the learner. We want to give you a quick but solid understanding, those aha moments, right? Without just drowning you in jargon.
Digestible.
Exactly. This deep dive is especially crafted to help you grasp the science behind skin health, which is relevant for everyone, whatever your skin type.
That's crucial. Skin health is universal.
Now, before we dive in properly, a quick heads up on pronunciation. In the Cütie Catcherz series, when we say cooties, we want you to think cooties.
Ah, like the childhood thing.
Yeah, exactly. Like the old playground term. It really fits the sort of playful tone and the metaphorical way the story works.
That's a great way to frame it. Makes it immediately relatable, doesn't it?
Right.
Bridges that gap between the science and the story.
Totally. Now, let's talk sources. Today, we're digging into a research paper. It's titled, Characteristics of Biofilm-Forming Ability and Antibiotic Resistance of Cutibacterium acnes and Staphylococcus epidermidis from Acne Vulgaris Patients.
Okay, quite a title.
It is. It was published in the Clinical, Cosmetic, and Investigational Dermatology Journal. The study was led by Dr. Kartika Rukyatan over in Bandung, Indonesia.
Oh, Indonesia. That's important context, actually. Microbial characteristics, resistance patterns,
Yeah.
they can sometimes vary quite a bit geographically.
Good point. So our goal today, it's really to uncover how the Cütie Catcherz comic series cleverly mirrors specific scientific ideas about C. acnes, especially how it behaves regarding those sticky biofilms and antibiotic resistance.
Connecting the dots between the lab bench and the comic book panel.
You got it. We want to equip you with a clearer understanding of what's happening on your skin and also how science can be the spark for these really compelling stories. So let's jump in.
Sounds like a really fascinating connection to explore. I'm intrigued.
Great. First things first, let's introduce our main characters, microbially speaking, Cutibacterium acnes and Staphylococcus epidermidis.
Ah, yes, the key players. These two bacteria are definitely central figures in Acne vulgaris, or AV, which, as the paper notes, is a super common and often pretty long-lasting skin condition.
Right. And this study specifically zeroed in on these two because, well, the researchers pointed out they're the most dominant bacteria found in AV, and crucially, both can form biofilms.
That biofilm-forming ability is key. Their prevalence and that ability makes them central to figuring out how acne develops and also how we might treat it effectively.
And this ties right into Cütie Catcherz. The cooties, as we're calling them in the comic, primarily represent C. acnes, right? They're the main sort of troublemakers.
The antagonists of the skin story.
Yeah. But it's interesting to think that maybe some other characters, or even just background stuff in the comic, might be subtle nods to other skin microbes, like S. epidermidis.
That's a very insightful way to look at it. Even if the narrative focuses on one main cootie, it can still subtly reflect the broader microbial community that's always there on our skin, like supporting characters.
Now let's get into the sticky part. Biofilms. The paper gives a pretty good explanation.
Yes, biofilms. Essentially, they're structured communities, microbes getting together, attaching to a surface, and they get embedded in this goo they make themselves,
Yeah.
an extracellular matrix.
goo they make themselves. Sounds cozy for them, maybe not for us.
Well, it's protective for them. Think of it like a group effort. The bacteria aren't just floating around solo. They're organized, shielded within this sticky framework.
And the researchers in this study, they reference earlier lab work, right? In vitro stuff.
Experiments done outside a living organism, like in a petri dish.
Right. And that earlier work suggested bacteria inside these biofilms can be way tougher to treat with antibiotics, like 50 to 500 times more resistant.
That's a huge difference, a massive increase in resistance.
Yeah. The paper even uses this analogy of a tiny sticky fortress. Really paints a picture, doesn't it?
It's a perfect analogy. That matrix, that goo, it acts like a shield. It just makes it physically harder for antibiotics to get in and reach the bacteria hiding inside.
Okay, and here's where the Cütie Catcherz link gets really cool. The comic actually features biofilm-fortresses, or sometimes cootie hives. It's where the main bad guy, King Cootie, hangs out.
Ah, so the comic visualizes the science.
Exactly. These structures in the story are all about protection, keeping the cooties safe, making them harder to beat. It directly mirrors the science of real biofilms.
That's a very clever visual metaphor, taking a complex biological behavior and turning it into something, really understandable and engaging for the story. A literal fortress.
So we've got the microbes, we've got their sticky fortresses. What exactly were the researchers in this study trying to figure out? What was the big question?
Okay, so the study's main goal was to investigate whether the biofilm-forming bacteria they actually found in acne patients showed different levels of antibiotic resistance compared to the bacteria from the same patients that didn't form biofilms.
Ah, so comparing the fortress builders to the non-builders and real people.
Precisely. They wanted to see what was really happening in clinical cases of acne, not just in lab dishes. They looked specifically at C. acnes and S. epidermidis, isolated from patients at Dr. Hassan Sadiqian Hospital in Bandung, Indonesia.
Studying them right from the source.
Which is a really important distinction, right? Studying bacteria taken directly from patients gives us much more relevant information about the clinical reality of acne and resistance.
Makes sense. All right, so how did they actually do this? What were the methods? Take us into the lab a bit.
Sure. It involves several key steps. First, sample collection. They got samples from closed comedones. Those are the early, non-inflamed bumps like whiteheads on the patient's foreheads.
Okay. How?
They use a neat technique called skin surface biopsy, SSSB. It involves using cyanoacrylate glue, basically.
Super glue on faces.
Gently. It's used to lift off the very top layer and the contents of the hair follicle. It's quite a clever, targeted way to collect the material right from the affected spot.
Wow, okay. Targeted glue lifting. Got it. Then what?
After getting the samples, they grew the bacteria in the lab, cultured them to identify the C. acnes and S. epidermidis. Once they had these pure isolates, they did a biofilm formation assay.
Assay. Fancy word for tests.
Essentially, yeah. They grew the bacteria in special plates under specific conditions. Importantly, C. acnes needs an oxygen-free anaerobic environment, so they replicated that. Then they measured how much biofilm each bacterial isolate actually produced.
So they could sort them, like good fortress builder, not so good.
Exactly that. They classified them as either biofilm-forming, BF, or non-biofilm-forming, NBF.
BF and NBF, okay. And the final step, the resistance check.
Right. Antibiotic susceptibility testing. They used a common method called disc diffusion. You put little paper discs soaked with different antibiotics onto a plate where the bacteria are growing.
And see if the bacteria grow near the disc.
Correct. If there's a clear zone around the disc where bacteria can't grow, it means the antibiotic is effective. If they grow right up to it, they're resistant. They tested 7 common acne antibiotics this way.
7 key tools. Which ones?
They used tetracycline, doxycycline, clindamycin, erythromycin, azithromycin, levofloxacin, and cotrimoxazole.
A good range.
Definitely. So by testing these against both the BF and NBF types of those bacteria, they could directly compare their resistance levels.
Okay, so we understand the setup, the methods. Now for the big reveal. What did they actually find? What were the results?
Well, they found some interesting things. First, just in terms of prevalence, they saw that biofilm-forming C. acnes were actually more common in the patients they studied. But for S. epidermidis, the non-biofilm-forming ones were more frequent.
Huh. So different tendencies between the two bacteria in this group.
Seems so in this population. But the most significant result, the real headline, was that contrary to what you might expect from those earlier lab studies, The researchers found no statistically significant difference in resistance.
Wait, say that again? No difference?
No statistically significant difference in resistance to any of the 7 tested antibiotics between the biofilm-forming bacteria and the non-biofilm-forming ones. And that was true for both C. acnes and S. epidermidis.
Wow. Okay. So in this study, building that sticky fortress didn't actually make the bacteria significantly more resistant to those specific antibiotics. That feels counterintuitive.
It is a noteworthy finding, yeah. It really suggests the relationship between making a biofilm and resisting antibiotics might be more nuanced, more complex than we maybe thought, at least in this real-world clinical setting with these drugs.
But did they find any resistance?
Oh, yes. They definitely found resistance to certain antibiotics, but it was present in both groups, the biofilm formers and the non-formers. Specifically, they saw resistance to clindamycin, erythromycin, and azithromycin.
Okay, so resistance to those three was an issue regardless of biofilm status in this study.
That's right, which is still important clinically. But the flip side was also good news.
What was that?
They found no resistance at all to tetracycline, doxycycline, levofloxacin, and cotrimoxazole in any of the groups they looked at.
Ah, so those ones still seem to be effective based on these findings. Okay, so how does this connect back to Cütie Catcherz? We've got King Cootie in his seemingly impenetrable biofilm fortress.
Well, this is where the science adds a really interesting layer to the metaphor, right? While the comic vividly portrays these fortresses as super tough, this research suggests that maybe antibiotic resistance is more complicated than just the fortress itself, at least for these particular antibiotics. Meaning perhaps other bacterial characteristics, things beyond just building the biofilm, play a really significant role in whether they can resist an antibiotic.
So maybe King Cootie's strength isn't just his sticky fortress. Maybe he's got other tricks up his sleeve, other defenses.
Precisely. It adds depth to the villain, doesn't it? His resilience might come from a combination of factors, maybe inherent genetic resistance, maybe ways to pump the antibiotic out, not solely the physical barrier of the biofilm.
Huh. The comic could even play with that idea. Like maybe there are different kinds of evolved cooties that are just naturally tougher, fortress or no fortress.
Exactly. It makes the story potentially more layered, reflecting the biological complexity even within a fantasy setting.
That's really cool. It feels more sophisticated.
Now, it's always important to put one study in context. What do other scientific studies generally say about this link between biofilms and antibiotic resistance?
Right. The paper does mention that the general idea, the concept that biofilms increase resistance, is pretty well established from lots of that in vitro lab-based research.
Exactly. That's the standard thinking based on lab conditions. But this study really highlights that what happens in a controlled lab setup might not perfectly mirror the complex reality inside a human hair follicle during an acne breakout.
Yeah, the real world is messy.
It is. And the authors also point out that other studies haven't always agreed on how common C. acnes and S. epidermidis biofilms actually are in acne patients. Some find them a lot, others less so. There's variability.
So the science is still evolving on that prevalence part too?
Mm-hmm. And the researchers here emphasize that their study is novel because they directly compared resistance between the BF and NBF strains of both bacteria, specifically taken from acne patients. That head-to-head comparison in this specific clinical context hadn't been done quite like this before.
And what about other bacteria? Does this finding line up with anything else?
Interestingly, yes. They mentioned that this lack of a strong correlation they found actually lines with some research on other bacteria, like Staphylococcus aureus and Pseudomonas aeruginosa. In those cases, too, the relationship between forming a biofilm and resisting antibiotics isn't always a simple, direct link.
So it might be a broader biological theme that biofilms are important, but maybe not the only key to resistance for many bacteria.
It certainly suggests that, yes, it's likely more complicated.
So let's talk real-world impact. What does this mean for understanding and treating acne, especially for you, our listeners?
Okay, well, the finding that there is resistance to clindamycin, erythromycin, and azithromycin, even if it wasn't linked solely to biofilms here, definitely reinforces why doctors need to be careful and judicious when prescribing these. Overuse leads to more resistance.
Right. Use them wisely.
Exactly. And the fact that this resistance was seen in both BF and NBF bacteria suggests that maybe just targeting the biofilm structure itself might not be enough to solve the resistance problem for these drugs. Other strategies might be needed.
But the good news stood out too. Tetracycline, doxycycline, levofloxacin, cotrimoxazole looked effective in this study.
Yes, and that provides really valuable information for guiding treatment choices. It also just underscores that acne treatment is complex, isn't it? It's not just about killing bacteria. It's managing inflammation, oil production, the whole skin environment.
And bacteria aren't monolithic?
Not at all. This study is another reminder that different strains exist, they behave differently, have different resistance profiles. It's not one size fits all.
And it's so important for all of you listening to grasp this, especially if you have darker skin. Acne affects everyone, and having a clearer picture of the science underneath the bacteria, the resistance issues, can really empower you. It helps you ask better questions, make more informed decisions about your skincare, about the treatments you choose. Research like this adds to that broader understanding for all skin types.
Knowledge really is power here. Understanding the why behind acne complexities can lead to much more effective, personalized ways to manage it.
Okay, let's bring it back full circle to our creative connection. How does Cütie Catcherz, with its cooties and fortresses, really work as a metaphor for all this science?
I think this series does it brilliantly. It uses the real science as a jumping-off point. The cooties are just a fun, engaging way to personify C. acnes, maybe even other skin bacteria, makes them characters you can visualize.
Instead of just invisible microbes.
Right. And the biofilm fortresses. That's such a direct visual translation of the actual biofilm structures. It makes a potentially tricky biological concept feel immediate and exciting within the story.
And what about the idea in the comic of those resistant, evolved cooties?
That maps perfectly onto the real-world problem of antibiotic resistance that this study touches upon. And interestingly, the study's finding that resistance wasn't just about the fortress. That adds a cool twist.
How so?
Well, maybe in the story, the resistance of the cooties comes from other things too, like specific mutations or special abilities they've developed. Makes them even more formidable opponents, perhaps.
I love that. Adds layers. And the comic also mentions things like hormonal havoc zones and how stress affects the cooties.
Yes, and that aligns perfectly with our scientific understanding that environmental factors, including our own hormones, our stress levels, can absolutely influence bacterial behavior, including biofilm formation. The comic is weaving these real biological influences into its world building.
Makes the fictional world feel more grounded somehow.
It does, and it's not just about, say, one big battle in the comic, is it? You mentioned a move towards more targeted strategies.
Yeah, it seems like the catchers don't just try to wipe everything out.
And that reflects the real scientific push, right? Moving towards more judicious antibiotic use, exploring other approaches that maybe don't just kill bacteria broadly, but perhaps target biofilm formation specifically, or interfere with how bacteria communicate, or maybe bolster the good bacteria.
Like precision strikes instead of carpet bombing?
A good analogy.
Yeah.
And even the idea of different types of cooties in the story could easily reflect the known genetic diversity within actual C. acnes strains, which the research also points towards.
So the comic really is using these metaphors to make pretty complex biology feel accessible, relatable, even fun.
Precisely. It's a fantastic example of science communication through creative storytelling, educating and entertaining simultaneously.
This study definitely leaves us with some lingering questions, though, doesn't it? Like, okay, if biofilms didn't significantly boost resistance to these antibiotics here, why did all that earlier lab work suggest they would?
That's a great question. And the answer is probably complex. Maybe those highly controlled lab conditions just don't fully capture the dynamic, messy environment of actual human skin with all the other molecules and cells around.
Maybe the fortress isn't as impenetrable in real life.
Could be. Or maybe other resistance factors just overshadow the biofilm effect for these specific drugs in patients. Things like genetic mutations in the bacteria themselves, or mechanisms they have to actively pump antibiotics out of their cells.
Like little bilge pumps getting rid of the antibiotic.
Kind of, yeah. Or maybe interactions with other microbes in the skin microbiome play a role we don't fully understand yet. It's complex territory.
It really makes you wonder what future research will uncover about all these intricate interactions happening on our skin. Maybe some of you listening will even be inspired to dig deeper, look into acne treatments, the microbiome, maybe even track down Cütie Catcherz and see how the science plays out there.
That's the exciting part of exploring these topics, isn't it? One answer, or non-answer in this case, just opens the door to so many more questions.
So let's try and wrap this up. What are the key takeaways from our deep dive today?
Okay, I think the main points are, 1, biofilms are definitely real. C. acnes and S. epidermidis make them in acne. But 2, this specific study in Indonesia found that for several common antibiotics, making a biofilm didn't automatically mean significantly higher resistance.
But resistance is still an issue.
Yes, absolutely. 3, Resistance to drugs like clindamycin, erythromycin, and azithromycin is a concern, seen in both biofilm-forming and non-forming bacteria in this study. That highlights the need for careful treatment choices. And 4, the Cütie Catcherz comic series is a really cool example of using these scientific ideas, biofilms, resistance as metaphors to tell engaging stories.
So hopefully this deep dive has given you, our listener, a clearer, maybe more nuanced view of the science behind acne and how it can spark creativity. We hope you feel a bit more empowered with knowledge about your skin health.
And it just shows how science isn't confined to labs, right? It pops up in culture, in art, in comics.
Absolutely. It encourages us to look at the world, even our own skin, with a bit more curiosity.
Well said.
Okay, finally, as always, here's a thought-provoking question for you to chew on. Given that this study didn't find that expected strong link between biofilms and resistance for these particular antibiotics, what other survival strategies might these tiny skin residents be using? What other tricks might C. acnes have up its sleeve in the complex world of our skin? And how could understanding those strategies spark even more innovative solutions, both in science and maybe in the next chapter of a story like Cütie Catcherz?
You got the cooties, we got the cure, fighting breakouts with facts and a whole lot of lore. It's Cütie Catcherz, beneath the skin, where science and story are about to begin. Let's go! You got the cooties, we got the cure, fighting breakouts with facts and a whole lot of lore. It's Cütie Catcherz, beneath the skin, where science and story are about to begin. You got the cooties, we got the cure, fighting breakouts with facts and a whole lot of lore. It's Cütie Catcherz, beneath the skin, where science and story are about to begin. Let's go!
That's a wrap on this episode of Cütie Catcherz Beneath the Surface, where science meets art and technology, and skincare gets a superpower treatment. If you learned something, laughed a little bit, or just loved the blend of art, culture, and biology, make sure to follow the journey at cutiecatchers.com and stay up to date on Instagram at Cütie Catcherz. Catch every new episode on Spotify and YouTube. Just search Cütie Catcherz Beneath the Surface. And remember, the cooties are real, but so is your power to fight back with knowledge. This is produced by me, Stephen Christian, of Stuck on an Island, in collaboration with Iltopia.